Good evening, ladies and gents and hermaphrodites. Welcome to the most prestigious, the most vermiculturally significant event of the year. Yes, I am talking about Mix Uniworms. Tonight, let's meet the contenders, the slithering sensations, the elegantly elongated worms. First up, from the Inoplian Empire, a worm with the elegance of a really elegant worm. Give it up for Mix Inoplea.
Good evening, the Uniworms rather. I am Capilla Ariella Arida, representing the class of ancestrally diverged nematodes, Inoplea. Our signature look is nothing like you've ever seen. We try to keep it classic with our cylindrical or bottle-shaped esophagus packed with 3 to 5 esophageal glands. We also have more stylish and more classy pocket-like amphids, and lastly, we keep a more polished style by having a smooth or finely striated cuticle. Generally, our life cycles vary by species. Some are passed in feces and then mature in the host intestine like Trichuris. Some may be found in undercooked meat with tiny cysts which then grow in the gut and move to muscles like Trichinella, while some are more complicated like us, Capillaria, that can spread directly or need an intermediate host like an earthworm or fish to end up in different parts of the host. Before we dive right into the class Inoplea, let's first differentiate these terms that we will use as we go along the worms of the Uniworms. Hermaphroditic refers to organisms that have both male and female reproductive organs. A good example of this is the invertebrate model Caenorhabditis elegans. Parthenogenetic refers to a female that is able to produce eggs or larvae without males in a process called parthenogenesis. This is seen in females of S. stercoralis. Oviparous refers to organisms that lay eggs that hatch outside their bodies, such as Trichuris trichura. Lastly, ovoviviparous refers to organisms that lay eggs containing fully formed embryo that hatch out immediately, like in Trichinella spiralis. Now, let me introduce class Inoplea. First, yours truly, Capillaria species. Us Capillaria are slender, hair-like nematodes with long steichosome-type esophagus, and we have eggs that are thick-walled and barrel-shaped with bipolar plugs. The males of our species are smaller with single spicules, while females are larger and responsible for egg production. Depending on the species, we can infect different organs such as the liver, respiratory tract, and intestine. Let me now introduce you to the other members of our class. First, we have Trichuris trichura, which unlike Trichinella, has egg, larva, and adult life stages. The eggs of Trichuris feature prominent polar plugs at either end. The adult forms both have a thin anterior end, but the female adult has a straight posterior end, while the male has a coiled posterior end. Next, we have the smallest human nematode parasite, Trichinella spiralis. Their larvae encyst in striated muscle tissues, and they have surrounding nerve cells that help in their dormancy and infectivity. The adult anterior heads are slightly rounded with a tiny stylet. They also have esophagus with a glandular steichosome made up of stichocytes which aid in digestion and nutrient absorption. Males have specialized claspers used for mating, and females are ovoviviparous and their vulva is found near the esophagus where developed larvae are ready for hatching.
Next, from the Chromatorian Collective, a contestant who's sure to make your blood thrill. From the depths of the digestive tract, please welcome Mix Chromadoria.
Hailing from the class Chromadorea with bulbous esophagus and amphids shaped like slits, pores, coils, or spirals. I am Ancylostome margimoran, and I stand by the saying, don't let the size fool you. I may be small, but I have a big bite. We hookworms are known for our, hooks. Specifically, our buccal capsules are equipped with sharp cutting teeth or plates, which vary among species, making it easy to attach to the intestinal wall. And we are dioecious. The ladies have a relatively straight posterior end, while the gentlemen ones are coiled and have a special feature called a copulatory bursa, or a fan-like structure used during mating. Our life cycle begins with an egg passed in the stool. From the egg, our rhabditiform larva hatches under favorable conditions and can grow and live freely in the same feces or contaminated soil. Then it develops into the third stage larva called filariform, which is now the infective stage. And these larvae enter humans by penetrating the skin, typically through bare feet, and are carried into the bloodstream to the heart and lungs. But it doesn't stop there. They ascend the bronchial tree to the pharynx and are swallowed again. And only then we can reach the small intestine, where we reside and mature into adults. Now, I'd like you to meet my dear cousin, Necator americanus. Our life cycle is pretty much the same. It's not infected by the oral route, but through direct skin contact. And we have identical looking eggs and larvae, but we differ more once we mature into adults. Because that's life. Unlike me, an Americanus possesses cutting plates instead of teeth and a copulatory bursa with fused spicules at the end.
Let me take over first, Ancylostoma. So, let's get on to the differences. Ancylostoma duodenale is larger while Necator americanus is smaller. A duodenale's head is bent parallel to the body, giving it a C shape, while N americanus has a curved head opposite from the body, so it looks like an S. Moreover, A duodenale has sharp teeth in its buccal capsule, while N americanus uses its cutting plates to attach to your intestines. Next, A duodenale's copulatory bursa is circular and the spicules are unfused, but they are fused distally in N americanus, who has an oval-shaped copulatory bursa. The caudal spine can be found in A duodenale but is absent in N americanus. And finally, A duodenale's vulva is post-equatorial, located about 1/3 from the posterior region, while N americanus's vulva is pre-equatorial, located in the middle of the body. But wait, I'm not yet finished. That's cause A duodenalis got a whole clan ahead of it. Meet the siblings, A caninum, A steylanicum, and A brasiliense. And of course, we can't forget Necator americanus there as well. But don't fret, all you really have to look at now are just the mouth parts and copulatory bursas, with the most prominent features labeled for you.
Another relative of mine is Trichostrongylus, which are similar to hookworms, but their eggs are much larger and have slightly tapered ends. All subsequent life stages like rhabditiform and filariform are similar to hookworms, but their adult forms lack the characteristic mouth parts such as teeth or cutting plates. Now we have our very own rat lungworm, Angiostrongylus cantonensis. Their eggs hatch and give rise to the first stage larvae in pulmonary arteries of the rat host until they are passed out in the rat's feces as the third and infective larval stage. Humans are accidental hosts by eating the intermediate hosts of Angiostrongylus cantonensis. In humans, they don't reach reproductive maturity but only up to the L4 and L5 stages. And the last member of my class, Strongyloides stercoralis, a highly adaptable nematode known for her ability to switch between free-living and parasitic life cycle. Her life cycle is different from other soil-transmitted worms. Unlike most nematodes, her rhabditiform larvae hatch directly in the intestines. These larvae can either be excreted in the stool and develop into free-living adults or transform into the infective filariform larvae with the ability to penetrate the skin upon contact.
And now, from the Ascaridoidea alliance, a worm with the unique ability to thrive in various environments, let's welcome Mixed Ascaridoidea!
Behold, from the illustrious Ascaridoidea, I, Gloria Diaz Caris, grace you with my presence. I am a dioecious worm with an esophagus that is triradiate and with prominent amphids sensing the very essence of my environment. My eggs are quite distinctive. The fertilized ones are rounded with a thick shell ensuring the survival of the next generation. The unfertilized eggs, longer and thinner shelled, are filled with granules. And this is me in my larva form. In my adult form, the difference between males and females is easily observed. And did you know in my adult form, you can tell the difference between males and females by their cross sections? The females, with their large egg-filled uteri, are quite prominent, while the males have smaller, more coiled reproductive tubes.
Next, from the Filarioidea Federation, a worm with a circulatory charm. Give it up for Mixed Filarioidea.
All the way from the worldwide superfamily of filarial worms, I am Mukerapia Wurzbach-Rofty, confidently representing Filarioidea. We are the skinny, elongated, thread-like nematodes specialized to invade and thrive inside the bodies of our vertebrate hosts, particularly in the lymphatics like in my case, and the tissues and body cavities for other filarial species. Be more careful with us because even with our slender bodies, our filarial infections can impact hugely like an elephant, such as in elephantiasis, which I cause. Also, our life cycles need only two hosts, a vertebrate definitive host and a blood-feeding arthropod intermediate host. We filarial worms also have distinct life stages, with our females being ovoviviparous. Once they inhabit the tissues of their definitive hosts, they hatch within their uterus the larva or microfilaria, which would further circulate in the blood and tissues of our definitive host. Then, the blood-feeding arthropod proceeds to take its blood meal on the definitive host, which would then carry the lucky microfilaria. Inside our intermediate host, the microfilaria develop into the infective larva that can then be transmitted to a new definitive host body during the arthropod's next bite. A fascinating thing about our microfilaria, some of ours exhibit periodicity, which means some ensure that their microfilaria appear in the host's blood at night. to align with the arthropod's feeding habits. Such an elegant system, if you ask me. Then, as adults, we filarial worms are dioecious, with females being generally larger than males. Now, we have mixed Brugia malayi, gracing us with their presence straight from Southeast Asia, bringing elegance, elephantiasis, and lymphatic filariasis. Transmitted by Mansonia and Aedes mosquitoes, one tiny bite is all it takes to join her fan club, though membership comes with a heavy price. Her microfilariae are sheathed with gracefully separated nuclei that stop just before the tip of the tail, which is an easy way to tell her apart from other filarial stars. And let's not forget her adult form, residing in the lymphatic system, where she and her dioecious partner block lymph flow, leading to swelling in the limbs. Now, let us compare two close relatives, Wuchereria bancrofti and Brugia malayi. While they share similarities, they have distinct differences. These differences include their size, cephalic space, nuclei arrangement, and terminal nuclei presence. However, they share the same infective, transmission, and diagnostic stages. So, while these two filarial nematodes share key life cycle stages, their size and anatomical features provide us with key differences. Those differences are very important when trying to diagnose which type of filarial nematode is causing an infection. Now, we have Miss Loa Loa saying helloa from Africa to your eyes. They are notorious for being the African eye worm. Watch out because you might just get this worm in your eyes from microfilaria infected chrysops flies with just one bite. Notice how the nuclei extend to the tip of their sheathed, tapered tail during the microfilaria stage. Here is Loa Loa as an adult with elevations on her cuticle known as bosses. Indeed, she is a boss, and quite an eyesore at that, literally. Next, we have Onchocerca volvulus. Their vector, which is the black fly, like Simulium species, can take a blood meal in the skin. Then, L3 larvae can enter, forming nodules, and eventually produce microfilariae. This can lead to a disease called onchocerciasis. Just remember that we only differ in our chosen habitats and the vectors that carry us. Moving on, let's talk about Manzanella species. Unlike other filarial worms, Manzanella microfilariae lack sheaths, and infections are often asymptomatic or result in mild symptoms, contrasting with the severe diseases caused by other filarial worms. The adult forms reside in connective tissues or body cavities, while microfilariae may circulate in the blood or skin. Last, for Filarioidea, we have the dog heartworm, where dogs, of course, are the main definitive host, and mosquitoes are the intermediate host. Their life cycle is the same as mine, with their ultimate destination being the pulmonary arteries and sometimes the heart. Males are smaller than females and exhibit a coiled appearance during mating.
And finally, from the Oxyroidea order, a worm that's always on the move, let's welcome Enterobius vermicularis.
Representing the order Oxyroidea with a double bulb esophagus and elegant cephalic alae, I am Enterobius vermicularis. We pinworm are among the most common intestinal parasites of humans, and our features make us easy to recognize. We are dioecious, where males come equipped with a single spicule for mating before dying shortly, while the females take one final journey to lay eggs, securing our legacies before one final bout. Females are about 8 to 13 millimeters, while males are shorter, around 2 to 5 millimeters, with a curved posterior end. One of our most notable characteristics is our pointed tail, which gives us our name, vermicularis, meaning worm-like. Our life cycle begins with the eggs laid in the perianal region, most often at night, causing enterobiasis. Infection occurs through self-inoculation, where eggs are transferred to the mouth by contaminated hands or by exposure to eggs in the environment, contaminated by surfaces, clothing, bedding, etc. Once swallowed, our eggs hatch in the small intestine, developing into larvae that move to the cecum and colon, where we become adults. This is what our eggs and larvae look like. Our egg is oval and asymmetrical with a thick, colorless shell which protects the developing larva inside. These eggs are highly resistant and can survive in the environment for extended periods. The larva shown on the right possesses a bulbous esophagus, a defining characteristic for our species. And finally, this is how we look like in our adulthood. The image on the left showcases the long pointed tail, which is a key characteristic in females. As mentioned earlier, the male is shorter with a curved posterior end. The key features would involve the double bulb esophagus, cervical alae, and a single retractable spicule.